Okay, alright. So, we're now down to the third part again of our video for Enterobacteriaceae, biochemical testing. I know medyo daghan na to so far. Gasakit na po na akong throat. Alright, but again, laban ta. Okay, alright. So, hopefully this is the last part na good of the vid. Okay, alright. So, we'll now continue with the next test, which is your triple sugar iron, diba? TSI. Na-mention na po ni sa culture media prep, right? Triple sugar iron. It's one of the defining tests to differentiate the members of the Enterobacteriaceae from other gram-neg rods, because we look into the different ability of your Enterobacteriaceae to ferment different types of sugar. Okay? Alright, and our indicators, number one is of course for pH, is phenol red. For the H2S production is ferric ammonium citrate, which is the source of H2S. But again, ang sodium thiosulfate ay ang source sa sulfur. But again, since diba H2S as a gas, dili man siya makitaan. So we add ferric ammonium citrate para na a black precipitate na ma-produce, okay? And and again, diba, as mentioned, TSI determines the ability of the organism to ferment different sugars, okay? And also the production of H2S, okay? And the carbohydrates here are sucrose and lactose at 1% ang concentration and glucose at 0.1%. Nganong gamay ra ang glucose? Because again, recall that all your Enterobacteriaceae are glucose fermenters. So, it's expected yun na medyo gamay ra ta sa glucose because wala na may pulos on identifying speciating them based on glucose fermentation, because we cannot speciate because all of them ferment glucose. So it's just like a confirmation if Enterobacteriaceae bagyod na imuhang organism. All right? Okay. So again, our principle, carbohydrates, fermentation releases acids that could change the color to yellow. Okay? All right. So and peptones or proteins found there in the presence of oxidation, right? Oxidation will now go to the red color, or produce amines that produce the red color. Okay. All right. So this is your TSI. That's again a butt and slant. So you have the slant or butt na deep. Okay. All right. For the slant, we determine if the organism is a sucrose or lactose fermenter. So ang sugars na nasa slant, lactose and sucrose. And for the butt, if yellow again, lactose and sucrose fermenter. That's letter A. We designate that as letter A. And red, K, non-lactose and non-sucrose fermenter. Now, how do I remember the letters? So you have A, Y. So A kung yellow, R, K. Red kung K. If red, kay K. A, Y, R, K. Okay? Alright, so again, if yellow, A, and if red, K. Okay, alright. And for the butt, again, we determine if it's a glucose fermenter. Still the same, yellow, A, glucose fermenter, and red, non-glucose fermenter. Now, there are some other mga results na to makita. If gas positive, it could be na ay cracks. Okay, mga cracks sa medium. Pwede na ay displacement, meaning musaka ang medium, as in naagyud. Okay? And we have also bubbles, okay, which again signify gas formation. And of course, H2S positive na ay black precipitate or blackening of the medium. Okay? Alright. So here's an example. Ayan, so for the first one, okay, diba? Slant na to is yellow, the butt is also yellow. So how do we interpret that? That's A over A. So this is the slant and this is the butt. As you can see po, nisaka ang medium. So it means gas positive. Okay? Alright, so A over A, gas positive, number one. Number two, still the same. A over A, H2S positive. Diba? As you can see, na ay black precipitate. And yellow na gina siya sa ubos. Okay? Alright. Okay. Ayan, A over A, H2S positive. Okay, number three. How about number three? That's K over A. As you can see, dili kay siya medyo klaro na yellow, but dili po siya orange. Ni-change na siya color. Maybe this is a late fermenter, okay? Or wala pa siya ni incubate yung intense. So K over A remains red ang sa slant and then yellow ang butt. Okay. Number 4, K over A pa rin. As you can see, di kay klaro, per na displacement, di ba, nisaka siya, gas and H2S positive. Grabe ang H2S positive, blackening yun. Alright. Okay. Alright. Okay. And number 5 is, of course, red ragyapon tanan, that's K over K. Okay. Alright, so I do hope nag-gets lang pag-interpret sa TSI. Okay? Alright. Ayan. Okay. So, we'll now go to some organisms that exhibit those results. Number one is, of course, A over A with gas, pero H2S nag is, of course, your rapid lactose fermenters. E. coli, Klebsiella, and Enterobacter. Remember, again, they are lactose fermenters and glucose fermenters, rapid pag yun. Yeah, because of their rapid lactose fermentation, they also produce gas, okay? So, muna na ay gas production. But they do not produce H2S. So, muna siyang EKE, Eke, E. coli, Klebsiella, and Enterobacter. Next, A over A, pero H2S positive, you have Citrobacter. Now, Citrobacter is a late lactose fermenter, remember? So, it's still positive kapon siyang A, yeah, glucose fermenter pag yun. And remember, appeal siya sa spaced. Okay, so Citrobacter. Okay, next, K over A, okay, non-lactose or non-sucrose fermenter, pero glucose fermenter, na ay gas, pero H2S positive, that is Salmonella and Proteus. Okay, because again, Salmonella and Proteus, they are non-lactose fermenters, but of course, they are glucose fermenters as they are part of the Enterobacteriaceae. And they also produce H2S, remember, spaced. Okay. K over A, H2S neg. Okay. K over A lang per H2S neg, it's your Shigella. Remember guys, Shigella is considered to be Shigella, biochemically inert. Okay. Biochemically inert. Like, inert. Daghan kayo siyang biochem. Nega kayo siya. Like, tanan niyang, if not all of its biochem is nega. Diba? Dili siya motil. H2S neg pag yun siya. Non-lactose fermenter. Daghan siyang nega. As in, daghan siyang nega ang Shigella. Basta gani, biochemically inert bacteria. Biochemically inert. Okay? It's your Shigella. Okay? Alright. Ayan. And for K over K, we have H2S neg. It's not a member of the Enterobacteriaceae. It could be Pseudomonas aeruginosa. Because Pseudomonas aeruginosa is an oxidizer. Okay? Dili siya fermenter. So, kailangan siya og oxygen in the presence, in fermenting glucose. Okay? Alright. And remember, our butt, di ba ang butt, glucose, it's really anaerobic ang environment. Sa butt, remember? Because again, walay oxygen na maabot dito. Okay? And Pseudomonas aeruginosa is an oxidizer. It really needs oxygen. That's why K over K siya. Pseudomonas aeruginosa, again, non-fermenter, not a member of the Enterobacteriaceae. Okay? Alright. Ayan. Now, next, we go now to the LIA, lysine iron agar. Actually, ang LIA, same po siyang principle in a way sa TSI. Okay? Pero different lang ang gina-detect na, breakdown substance or gina-utilize sa substance sa bacteria. For lysine iron agar, again, it's still a butt and slant. But again, it's based on the decarboxylation and deamination of proteins. Okay. Or of a lysine, di. So, that is a protein. Oh my gosh. Okay. Protein or, protein, of lysine, decarboxylation or deamination. And also H2S production. Now, for indicators, this time it's bromcresol purple. So, purple ni siya. Actually, very characteristic, very specific food for lysine iron agar. It's color purple. Because again, of bromcresol purple. If acid, yellow, alkaline, kay purple. Still the same H2S is ferric ammonium citrate. Sodium thiosulfate ang source sa sulfur. But again, para ma-manifest or ma-visualize atong H2S production, we add ferric ammonium citrate para mag-create of black precipitate. Okay. And the carbohydrate is only glucose. Because again, we're not after mostly on the carbohydrate utilization. It's more on the lysine, which is our amino acid, L-lysine. Okay. Now, we go now to the principle. Again, it could be either deamination or decarboxylation. Now, deamination happens in the slant because deamination requires oxygen. Okay? Remember, slant, but slant, di ba? So, ang slant, it's really exposed to the air. So, deamination really requires a lot of oxygen down. It's very aerobic ang reaction. Okay, so lysine, in the presence of lysine deaminase, kay deamination, muna ang products, ammonia plus alpha-ketocarboxylic acid plus ferric ammonium citrate and flavin mononucleotide, which is a coenzyme, which now releases a burgundy or Bordeaux. This is Bordeaux. Okay? Burgundy or Bordeaux red color. Okay, so therefore, ang positive sa imong deamination is red. Burgundy or Bordeaux. Bordeaux niya, Bordeaux. Diba? Social kayo. Bordeaux, red color. Okay. And for lysine decarboxylation, but it requires anaerobic environment. So lysine, in the presence of lysine decarboxylase na enzyme, it produces CO2 and cadaverine, which gives the purple color. So ang positive nato sa decarboxylation is purple color because of cadaverine. Please take note of this byproduct, cadaverine. Okay. All right. Ayan. So again, this is your lysine, slant and butt. So for lysine deamination, you have burgundy is color red. And ang negative na to is purple, K. All right. So again, quite the same rang ato ang pag-label pod or pag-report sa results, same sa TSI, remember? But for this, again, red is K, AR. Sorry. So, ayarog kalibog. Red, R, okay? And purple is K, PK. Alright. Okay, PK, deaminase, negative. Now, for the ubos, lysine decarboxylation, yellow ang negative, di ba? Remember? And positive ang K. Purple pa rin. So, PK, AY pa rin. So, AY, PK, para sa TSI na AY, RK, ang difference lang is sa lysine decarboxylation, negative ang yellow, positive ang purple. Okay? Alright. Because again of cadaverine. Alright? Please take note. Okay. And H2S positive na blackening of the medium. Alright. So here's example number 1. Unsa man ni siya? Okay, that's K over K. Number 2, purple, K over A. Alright? So decarboxylase, deaminase negative, decarboxylase negative pag yun. Number 1 is deaminase neg, decarboxylase positive. Okay. Number 3. Okay, so number 3, that's K over K, H2S positive. Okay, so as you can see, na ay blackening. Alright? So meron siyang sapin-sapin, guys. I don't know if they count mag-sapin-sapin, but it's very delicious. Pero yun yung itsura. Number 9, purple, na yung mga shades of different colors. And number 4 is, I don't know if klaro ang red, pero yeah, it's red, R over A. Okay? Alright. Okay, ayan. Alright. So we now go to different organisms pa rin. K over K, meaning deaminase neg, decarboxylase neg, decarboxylase positive, H2S positive. That's Salmonella. Okay? K over A, deaminase neg, decarboxylase neg, pero H2S positive. That is your Citrobacter. Okay? K over A, still the same for H2S neg, that's your Shigella. As you can see, dagan kayo siyang negative, no? Negative siya sa life, ang Shigella. Okay? And very characteristic yun, R over A. So, deaminase positive. Per decarboxylase neg, H2S neg, you have the PPM. Sila gina magsigkuyog. Proteus, Providencia, and Morganella. But note, Proteus, di ba, produces H2S. Remember? Appear siya sa space. But, ang LIA, it's not sensitive to its production. Okay? So, dili niya ma-detect ang production sa Proteus sa H2S. So, negative ragyapon siya sa H2S in this culture medium sa LIA. Okay? Alright. Ayan. Okay, now we go now to the next test. Actually, quite the same with principle with lysine LIA. This time, ang ginagamit lang is ornithine. So, decarboxylation of ornithine. Indicators na to is still bromcresol purple. And ang amino acid is ornithine. Now, for ornithine, in the presence of ornithine decarboxylase, it now results to the byproduct na putrescine, which gives again a purple color. Remember again, yung say byproduct sa lysine, cadaverine. Now for ornithine, it's putrescine. Now please take note, guys, kay usahay sa exams or sa boards mo yung na what is the byproduct of ornithine decarboxylation? Putrescine. For lysine, cadaverine. Okay, wala kay ko yung mnemonics ana, I forgot unsa, but I think letters siya ko ato like L, C, O, P, may anar ako. Okay, but again, maybe you will find much more effective mnemonics for your studying, so go lang, laban. Okay, so still the same for the next one is arginine decarboxylase or dihydrolase. Now, it could be two reactions, either of the two reactions. So again, decarboxylation or hydrolysis of arginine. Okay, indicator is still the same, bromcresol purple. Amino acid is arginine this time, and the principal arginine. In the presence of arginine, decarboxylase becomes CO2 and agmantine. Now, agmantine can still be broken down into ammonia and putrescine, which now gives the purple color. That's the first reaction. Now, arginine, in the presence of dihydrolase, lahi na enzyme, mo create siya og lahi na byproduct, phosphate, ammonia, and citrulline. Now, citrulline can be converted further or broken down to ornithine. Now, this will now follow the reactions of ornithine from the previous slide. So, ornithine decarboxylase, okay, will now give to putrescine, which is still a purple color. So, either way, purple color ang result. Okay? Alright. So, again, for ornithine and arginine, still the same. Left is positive, negative is yellow. Basta positive is the purple color. Now, for the next slide, I will tell you, or like asa siya magamit. Example for some organisms lang that could really differentiate them with each other. So number 1 is Klebsiella pneumoniae and Klebsiella oxytoca. Di ba remember, unsa na test ang maka-identify ra gud nila? It's indole. Kinsa indole positive? Oxytoca, na letter T, so positive. Siya indole positive. Now, as you can see, medyo same sila, di ba? Same sila, result ra pud sa LOA. LOA na siya, LDC, ODC, and ADH. So, wala siya pulos na test po usually. So, how do we, para asa ni siya, it's for Enterobacter and Klebsiella. Remember that Enterobacter and Klebsiella, they have the same MVIC profile. They have, ano yung MVIC profile na? Mark, okay. Alright, negative, negative, plus, plus. Okay? Alright, so, we now go to other tests para ma-isolate good sila. So, we could perform the decarboxylase or dihydrolase test. Now, remember, an Enterobacter aerogenes. Okay, A. So, how do I remember? Sa A ra siya negative. O, diba? Okay, Enterobacter aerogenes, sa A ra siya negative. Whereas ang Enterobacter cloacae, or cloacae, na L, sa L ra siya negative. O, diba? Okay? So, the rest positive na. O, so, easy ra to remember. Enterobacter aerogenes, sa A ra siya negative. Enterobacter cloacae, sa L ra siya negative. Okay? Alright. Now, for Pantoe agglomerans, very characteristic po niya, negative tanan. That's why if i-ask mo triple decarboxylase, okay? Decarboxylase negative or dihydrolase negative, it's Pantoe agglomerans. Okay? But kinsa ng triple decarboxylase or dihydrolase positive, it's Plesiomonas shigelloides. Ayan, wala ni gawa sa mong boards, but I was like na amazed ko pag nakabalo ko sa pag-study na ako, sa pag-review na ako. Alright? So again, triple decarboxylase negative, it's your Pantoe agglomerans. Triple decarboxylase positive, it's your Plesiomonas shigelloides. Okay? Again, Enterobacter aerogenes, sa A ra siya negative. Enterobacter cloacae, sa L ra siya negative. Okay? Alright. Ayan. And sa kanina test, ang maka-differentiate sa Kleb pneumoniae and Kleb oxytoca, indole. Wag na mag-isip. Okay. Alright. Next, phenylalanine deaminase test. Still the same principle. Ang difference lang is, again, the amino acid na ginagamit. Ang reagent this time, we use 10% ferric chloride. Okay? And amino acid is still phenylalanine. Okay? Ang principle, phenylalanine in the presence of the enzyme PAD, phenylalanine deaminase, will result to the production of NH3 and phenylpyruvic acid. Now, in the addition of FeCl3 sa phenylpyruvic acid, it will create a green color. Ayan. So, ang positive is green color. Ang phenylpyruvic acid mo combine with ferric chloride to produce a green color. Okay. Ayan. So, here's an example. Of course, ang positive na to is ang left, color green, and negative is right. Okay. And ang PAD, guys, we use there a slant. As you can see, slant na siya. So, para sa citrate slant, ang PAD pod kay slant. So, how do we inoculate? Streak. Okay, streak ra siya. Alright? Phenylalanine deaminase test. Okay, and our last, dili pa day. Okay, kinsa na mga PAD positive? Again, ang trio gyapon nato ang bestie sa lahat, ang Momshi, ang mga Momshis na magandang buhay. You have PPM, si Momshi Proteus, Providencia, and si Morganella. As you can see, as mentioned, sila gyud ang tulo magsigikuyog. As in like, trio gyud na sila. Trio tagapayo, sila yung mga besties. Alright, mga you can't sit with us. Okay, charot lang. Okay? Alright, so PAD, phenylalanine deaminase positive. And the last test for our Enterobacteriaceae, yay, okay, is of course the Urease test. Again, as mentioned, it's not only found in the Enterobacteriaceae. It can also be used to other bacteria. Di ba remember, unsa na test ang maka-differentiate sa C. diphtheriae, Corynebacterium, with others that can produce brown halo on CTBA, C. ulcerans, and C. pseudotuberculosis. It's the Urease test. Because again, Corynebacterium is, diphtheriae is urease negative. Okay. So again, detects the presence of urease. Atong indicator is phenol red. So in the principle, urea in the presence of urease converts it to H2O, CO2, and of course, ammonia. And ammonia is of course basic. Alright, so it will now give a bright pink or magenta color. So atong positive reaction is pink. Okay, and we could use either a Stewart's medium, which is a broth, or pwede po siyang Christensen's urea agar. So pwede siyang slant. Okay, so if broth gani, Stewart's medium. But if slant, Christensen's urea agar. And again, the positive result is pink color, bright pink or magenta color. And ang negative is light yellow. Okay? Alright. And kinsa ni mga positive na urease? Alright. Okay. Alright, so we have the rapid urease producers, meaning paspas kayo silang produce of urease, is of course the PPM. Proteus, Providencia, Rhett-Gary, and Morganella. Recall sa IMVIC nato, di ba? Providencia, Rhett-Gary, and Providencia stewarti, they have the same IMVIC profile. So, sir, saan na ito pag-differentiate sa duha? You could use urease. Because again, Providencia, Rhett-Gary ang positive sa urease. Providencia stewarti is negative. Okay? Ayan. Alright. So again, pero magkuyog na Japon sila, PPM. Proteus, Providencia, Rhett-Gary, and Morganella. And we also have what we call the late urease producers, or slow rather. We have Citrobacter, Kleb, Enterobacter, Gergovii, Yersinia, and Serratia. And our mnemonics for that is C keys. C keys. Ayan. Alright. C keys. Okay. Alright. C keys. Alright, so again, slow urease producer. Citrobacter, Kleb, Enterobacter gergovii, Yersinia, and Serratia. So these are slow urease producers. So dili siya inanakapaspas, pareha sa rapid urease producers. Okay. Alright, so basically guys, that's the end. Whew. Okay, take a deep breath. Inhale, exhale. Okay, so I advise after siguro ninyo maminaw ani na pre-recorded lecture, try to take a deep breath muna, no? Ipapahuway sa imong brain. Or eat something sweet para more glucose for your brain pa rin. And, you know, again, and contemplate kung ano kang gamit. Tech char, joke lang. Alright, again. So again, we're done with Enterobacteriaceae na biochemical testing. I know it's too much. Again, daghan kayong information to really digest. But again, try your best to really internalize and put into heart good. Maybe this is one of the reasons why some students find bacteriology difficult because again, there's a lot to really master, especially sa biochemical testing, the reagents, the positive reactions, and all that. But once you find really na magsimog tuon, magsimog repeat ani, bahalag sumod siya or like kapoy na jud. If ever mamaster na gyud ninyo, it's easy na lang yun, chicken na lang, and like chada gyud siya. Like it makes bacteriology really, really interesting, and that's why I love it. Good. All right, so again, that's all for Enterobacteriaceae. Again, TMI gid siya, no? Dagan kayo remember. Remember lang ato mga mnemonics. Don't forget those. Dagan na kayo. Medyo naglibog na po asa na to sila. But again, those can help you. Okay? And if you have other mnemonics put for you that you find much more effective kaysa sa akong na-share, then go, feel free to use it. Okay? Di man po required na gamiton ninyo akong mnemonics. But again, I'm just sharing lang kung unsa akong mga nagamit during review. Okay? Alright? Because again, Enterobacteriaceae, a lot, daghan dyan kayo nigawas sa imuhang amuang boards sa microbio. And they usually phrase it in questions as case study. Like muna siya, nigamit ko na specimen, kani ang biochem, like indole positive, IMVIC, muna yung IMVIC, citrate positive, motil, non-motil, inana. And choices na, unsay ang organism. So, inana na siya. Okay? Alright, so thank you so much, dears. I do hope na mo na-learn. Yeah, medyo sakit na po akong throat. But again, at least we're done with this topic. So again, if you have any questions, just feel free to chat me again in our group chat ba? Okay? And I'll see you on our next pre-recorded lecture on biochemical testing pa rin. Okay? So have a great day, guys, and keep safe.